It's really an honor to talk after such a highly distinguished speaker, and I hope that I will give you some good information as well. So Praxis Genomics has been in existence for about 4 years. We are located in Atlanta, Georgia, and we consider ourselves sort of a top-notch diagnostic laboratory simply because we report out structural variants that no other current company actually reports back out. And how can we do this? That's what I'm going to talk about. So about 50% of patients currently go without a real genetic diagnosis, even after extensive testing. And cancer samples or cancers of patients are even less well characterized. Fewer people get personalized treatment. So the goal of Praxis Genomics is really to provide genetic diagnosis for these difficult to solve cases. So we get all the cases that nobody else can solve. Not all, unfortunately, but we get a good number. And then we solve them mostly because we focus on structural variants and we do report those out. So I want to bring up a case, and it will sort of be the theme of my talk, sort of illustrate how our approach is superior to what's out there currently. And this is a case study. Uta Franke, who is a distinguished professor at Stanford and has been one of the greats of human genetics in this and the last century, she came to me, she's on our advisory board, and she came to me that she has a case that, she thinks it's definitely genetic and she couldn't solve it. Nobody could solve it for her, and if we take a try on it. So it's a 17-year-old male with supravalvular stenosis, failure to thrive, delayed development, short stature, acromegalia, dysmorphic features, hypertelorism, macrocephaly, syringomyelia, hypertension, and anxiety disorder. So they did FISH on this kid, they did CMA, they did exome, and they couldn't get an answer. So why is that these really sophisticated and used, highly used, widely used technologies fail to give answers? So for microarrays, the problem is the resolution. Unless it's specifically designed, the resolution is usually 10 kb or so for most of the genome, right? So they fail to detect also copy number variants that are, copy number neutral structural variants. So translocations, inversions are not detected. And they don't provide necessarily a precise position of the breakpoints. The targeted testing, both the panels and the exomes, these reports or these studies are just as good as your design of your capture and your ability to call, let's say, copy number changes on your captured exons, which is very tough, very difficult, and often not done. Clearly, they also miss repeat expansions, and often mitochondrial genomes are not included in the exome or the panel, even though it would be justified by the phenotype. So that's why we kind of propose, just go straight to genome. Don't do panels, don't do exome, don't do FISH, don't do a separate test, let's say, for FSHD or repeat expansion disorders. Just go straight to the genome and approach it from 2 points, 2 technologies, the short read technology and the optical genome mapping, which I'm going to talk about. So this is really our current paradigm. So karyotyping, microarray, repeat expansion testing, sizing, FSHD testing sort of can be pretty much replaced by BioNanoSapphire optical genome mapping. And then again, microarray, repeat expansion testing, sizing, panel sequencing, exome sequencing, mitochondrial genome sequencing, mitochondrial depletion testing, uniparental disomy. All these tests that you order independently or individually, they can be replaced by genome sequencing. And so you use simultaneously, preferably bio-nano-optical genome mapping and genome sequencing, and whatever you find, you verify with a transcriptome, if the proper tissue is available. So that's kind of our approach. And you say, okay, well, that's nice, but is it affordable? So the way we try to help with that is, first of all, we use the T7 machine, which allows us to offer pricing that nobody else can. And the other approach is that we basically do the genome sequencing, and then we report it out at a lower entry price as an exome. If you find the cause of the problem, you don't have to pay more. If you don't, then you can upgrade gradually to expanded exome and complete genome. But you only pay the difference between the costs, and you don't have to submit new sample. And if you still haven't found it, you can upgrade to OGM. And we always extract ultra-high molecular weight DNA, so we can just immediately do the optical genome mapping right away from the same sample you submitted originally. So it's a really nice way, very quickly upgrade if necessary, but you have a lower entry point into the testing. So this is this wonder that I have had the privilege to have in my lab since I think early summer or something. Yeah, early summer. And I was told when they installed it, you know, that you had sequencers before, but this one is really sexy. So I'm like, okay, well. So it's sexy in black, right? And it has a long name, like DNBSEQTM. So I just call it T-Rex because it's really fast and it's a monster. We had to disassemble the door of the lab to get it into the lab. It's exactly an inch wider than most American door frames. So you have to pay attention to that. But it's wonderful. And then the sales rep gave me this little plush animal representing T-Rex. But we love this machine. Before we had a problem with turnaround time because we had to wait up until we had enough samples to run an Illumina run, right? Illumina 6000 or other machines, even more samples you need to have. But with this machine, I can do a run when I have like 12 genomes, or if I want to eat the cost, I can run it pretty much, with less, because it's 4 independent sequencers inside this black box, and so I can start and stop any one of them independently. If one gets stuck, it doesn't mean that I can't touch the machine until one of the good runs finishes, flow cells finishes, and then have to rerun the second flow cell from scratch, right? I can just stop any one of them independently and start it again. So that's a huge thing. And the speed is really amazing. Overnight, right? I start the run, 12 o'clock, next day I have the sequence 12 o'clock. I upload it to the cloud. I have the analysis done. By next morning, I can sign out the report. So before it was like a 3-day kind of thing, 3-day for the run, and then the upload to the cloud. So it was more like a week. So this allows us to report out genomes really quickly. Currently we do like a 5-day, which is our sort of fastest, but we can do probably 3 days or so if it's really important. So that's kind of what we do. And of course, my other beauty, this one maybe not as sexy, I don't know, it's a personal choice, but this machine has been with my lab now for 4 years. I was one of the first one who bought this machine, and I was amazed by the technology. You can actually see, the DNA molecule, individual DNA molecules with a microscope, real-time floating by, So I love this machine. I'm not going to go into the details, but basically what it is, individual DNA molecules are labeled based on a 6-base pair recognition sequence, and that creates a unique profile for every DNA fragment. And then from this barcode, you basically assemble the whole genome de novo, and you analyze the changes based on the distance between the barcodes. So these are the kind of how it looks. The top is the reference, the bottom is your actual patient sample, and if the distance between the labels shrinks, it's a deletion. If it increases, it's an insertion, and you see the rest. If it's an inversion, the whole pattern flips over. Translocations are beautifully detected. I think the biggest thing about this machine that it makes it visually very easy to identify translocations, reciprocal translocations, and inversions. No other technology currently does that kind of visual output what OGM can do. And I think it's probably more like a software problem that is not solved by the currently available software company products. But they did solve it. And then we use Genox for annotation. It's an Israeli company. We had good luck with them. And they allow co-interpretation or co-annotation of data from the sequencing and the optical genome mapping. So I can really just view one screen. I use IGV a lot too. I always look at the native data, the actual coverage. That's so important. And so that's how we now I'm continuing with this case. Why that case wasn't solved. So you see here an IGV display of a duplication, 70 kb duplication on the distal P arm of chromosome 7. The proband is on top, the healthy brother is on the bottom, right? So there's a gene there called UNCX. Nobody knows anything about this gene. This was disregarded on microarray because they must have seen it. It's big enough so that they must have seen it, but they just didn't report it, they didn't care because there was so little information about it. And microarray, you don't know where that piece is. You don't know if that piece is in situ duplicated, is it head to head, tail to tail, or it's went to some other chromosome. You have no clue about that. And that's an important deficiency of microarray. So then we found also that in the proband, there was a deletion, about a 20 kb deletion on the X chromosome. And that involved the gene BRCC3, which is in a complex with BRCA1 and 2. And so that was interesting, There were some cases reporting out Moya-Moya syndrome with BRCA3 deletion. So I was thinking, yeah, this kid might have Moya-Moya syndrome, but then Uta said, no, but he doesn't have Moya-Moya syndrome. We tested him. We looked at the blood vessels. He doesn't have Moya-Moya syndrome. So interestingly, the optical genome mapping, instead of this deletion on the X chromosome, showed an insertion, right? So that's really strange. Sequencing shows a deletion, optical genome mapping shows an insertion. And I noticed that the insertion was actually, had the pattern of that duplicated region on chromosome 7. So these are the kind of things that you don't pick up with sequencing. Or, if you are very good if you pick up with sequencing data analysis, because there is so much noise and so much false positive chimeric reads because of the deficiencies of our reference, right? But here you go. It's pretty easy. You have a sequence, you have OGM, and then you kind of solve the case. So a piece of chromosome 7 is duplicated. The patient has 2 normal 7 chromosome, and then it gets translocated into the deletion of the X chromosome, right? And then you show that you do the transcriptome, and you show these are 2 controls, and you show that, yes, the expression of the genes involved in this deletion insertion is lost. So is it a novel syndrome? We think it is a novel syndrome. And this is the summary, really. So our study illustrates that OGM and whole genome sequencing can detect and localize structural rearrangements, copy number variations previously missed by standard of care testing. And that's the take-home message, really, that I can solve unsolvable cases by all the other labs because I look at structural variants and because I have an alternative way to prove that they actually are true. And no other lab can do that without an OGM, really. So reporting out structural variants is lagging because of that. And then transcriptome is great to verify the functional significance. And finally, the end is that optical genome mapping and whole genome sequencing is a powerful tool and likely to become recommended genetic testing in the future. And I hope that the future is now. So, you know, I think if you have unsolved cases that you would like to run through our lab, because of this special day, I offer an $888.88 genome and an $888.88 optical genome mapping for the first 50 people, you know, in the audience, or if you have friends that are interested, so that I can prove to you that this approach really is worth. And you wonder why 88888 is because I was up on Fuji Mountain in '88 on the 8th of the A. And so that's a special lucky number in Asian culture. And so since I found my good fortune with the T7, I'm happy to oblige to follow the Asian tradition about good luck. So thank you so much.